So good afternoon. It's 5 o'clock, so let's start today's MENDEL lecture. So good afternoon, everyone. I welcome you to this MENDEL lecture, and it is my pleasure to announce today's speaker, who is Professor Virginijus Šikšnys. And I will briefly introduce him. So Professor Šikšnys comes from Lithuania. He's a professor at Vilnius University. He actually holds several positions there. He's a chief scientist and head of a department at the Institute of Biotechnology. And he's also the scientific director of the Institute on Genome Editing at the Life Science Center at Vilnius University. And this is actually an EMBL partnership institute, as far as I understood. And Professor Šikšnys is biochemist by education, so he studied organic chemistry at Vilnius University, and he then moved to Moscow to do his PhD at Lomonosov Moscow State University, where he studied enzyme kinetics and catalysis. And he actually got some Czech connection there because his PhD advisor was a Czech researcher, Karel Martinek. And then he moved back to Vilnius, where he did his research at the Institute of Applied Enzymology until 1993. And in 1993, he decided to move to Germany. And as a visiting scientist, he was at Max Planck Institute for Biochemistry in Martinsried in Germany. And he actually worked there together with Nobel laureate Robert Huber, working, I guess, on restriction endonucleases at that time. And in 1995, he moved back to Vilnius University, to the Institute of Biotechnology, where he is the head of a laboratory. He's the professor, chairman of a Council of Life Sciences Center at Vilnius University. So Professor Šikšnys is member of EMBO. He's member of Lithuania Academy of Sciences, Norwegian Academy of Sciences and Letters. He published over 130 papers, and it seems, at least in the last period, most of the papers are in journals like Nature, Cell, Science, NAR, and so it's pretty impressive. Of course, he got several awards and prizes for his outstanding research. Like, for example, Warren Alpert Award in 2016. In 2017, he got the Novozymes Award from the Danish Novo Nordisk Foundation. He got also Lithuanian National Science Award. And finally, in 2018, he was awarded with a prestigious Kavli Prize, which he shared together with Jennifer Doudna and Emmanuel Charpentier. And if you look at his research, he actually spent, I guess, 2 decades working on restriction endonucleases at first. Until he stumbled upon a new defense mechanism that bacteria developed to fight against invading viruses. And he immediately realized the potential of this mechanism, and this is what we know today as CRISPR-Cas9. And so he started to work on this and apply it into genome editing technology. And Professor Šikšnys was actually one of the first researchers who discovered CRISPR-Cas9, and he published his findings along the same time as Jennifer Doudna and Emmanuel Charpentier. And he published several seminal papers on Cas9 biochemistry and laid foundations for translating the CRISPR-Cas9 system into this powerful genome editing tool. So while we, or most of us, work with CRISPR-Cas9 and use it as a tool, he actually works on CRISPR-Cas9, and not only Cas9, but also other enzymes and other mechanisms that bacteria have evolved to fight against viruses. And so that's all, and now the stage is yours. It's an honor to have you here. Thank you for coming. And before I give you the word, I would actually take this opportunity to hand over this MENDEL medal.
Thank you very much. Thank you. Thank you, Nisa. Thank you. Thank you. That's really great.
There is a certificate associated.
Thank you.
Yes.
Dobry wieczór. Thank you, Michal, for really kind introduction, and it's my great pleasure to be here, and I'm really thrilled to give a talk at the place where actually the foundations or key concepts of genetics were developed by Gregor Mendel. And there is another reason that I enjoy being here. So this is probably the only other place in the world where my family name is transcribed and pronounced correctly. And yeah, and including these strange epigenetic marks on the on the top that doesn't change the meaning too much. But anyway, there is a simple explanation, because Lithuanian nobles actually got studied at Charles University in Prague, and together with their diplomas, they also brought these kind of epigenetic marks into Lithuanian language, and we still use it. So it's really a nice Czech and Lithuania connection. And I don't know how many of you have been in Lithuania, but my lab is located in Vilnius, and Vilnius is the capital of Lithuania, one of the 3 Baltic countries. And Vilnius is an old city, probably most famous for the number of churches that probably, I would say, overcompete the number of churches in Brno. But it doesn't mean that we go to the church every morning. Just across the river there is a modern city, and in Vilnius history and modern times live peacefully together. And again, Vilnius University is one of the oldest universities in the region. It was established in 1579, and very recently, or not very recently, but some time ago, we moved into a brand new life science center where my lab is located. So there is another interesting commonality between Brno and Vilnius. And Gregor Mendel made his key discoveries here in Brno, in the outskirts of Empire. And his discoveries were forgotten for many years. And it took quite some time when they were actually surfaced and recognized. And Andrzej Sniadecky actually taught chemistry at Vilnius University, and actually he also made very important discoveries. For example, he introduced the term organic chemistry. Probably this was first mentioning of organic chemistry as a discipline in his book that was named the Theory of Organic Beings. And he also laid the foundations of biochemistry, but the problem is that his discoveries were forgotten and went into oblivion. And the reason is that actually he published his works mainly in Polish language, and that therefore his works were not available for the wider scientific community that spoke probably Latin at that time. But in fact, this example just shows that discoveries, the groundbreaking discoveries, happen in different places of the world, including Brno and Vilnius, and sometimes they become forgotten for different reasons, maybe lack of communication, wrong language to publish your work. But anyway, to pay the contribution to Sniadecky's, recently the book Theory of Organic Beings was translated into English, and now it's became available for wide scientific community. And I brought this book with me and would like to present this book for the organizers of the MENDEL lectures, and maybe it will find some place in the library. So Sniadecki actually had an interesting view on human nature. And in his book, he claimed that man has certain characteristic organic form by which we distinguish him from all other beings. And the important thing is that he postulated that man's existence and life depends on the organic force. And this force actually acts commonly and continually, as he wrote, on organic combinations contained in the body. It means on organs present in the body. And these organs actually process food and air and water that are essential elements of life. But he also made an important discovery, saying that sometimes this force also, not very often, accidentally acts on non-viable matter, including plagues and venoms. And this was kind of a first indication that interactions with environment are very important for human beings and our lives. And viruses were not known at that time, but I think that this would be a nice illustration that viruses can shape our life as human beings. And now we know that immune system in our body protects us against pathogenic microorganisms and viruses that are present in our environment. And it has two branches that are called innate and adaptive immunity. And the innate and adaptive immunity are comprised of different set of molecules and organs, and they differ in a response time. So the innate immunity reacts to the invaders immediately, while in the case of adaptive immunity, a time is needed to develop a response. And if our immune system fails, so then we lose our fight against the invaders. So bacteria, even the pathogenic ones, they have their own enemies. And bacteriophages, or viruses of bacteria, infect bacteria in order to replicate, and often kill bacteria when replication is completed. But as we know, bacteria still survive and thrive and multiply. And this is because bacteria during evolution develop antiviral defense systems that actually interfere with viral attacks. And these antiviral defense systems in bacterial genomes are clustered in a so-called defense islands. And in these defense islands, you can find restriction modification systems, CRISPR, and other antiviral defense systems. And taken together, they comprise a primitive immune system of bacteria that allows bacteria to find invaders. And in my lab, we aim to understand molecular mechanisms of antiviral defense provided by different antiviral defense systems, and we are using a combination of biophysical, biochemical, and structural approaches. So for many years, we were studying restriction modification system. And this is a simple system that is comprised of two enzymes, restriction endonuclease and DNA methyltransferase. And restriction enzyme actually is able to recognize short nucleotide sequences in the invading foreign DNA and cut, introducing a double-strand break. In fact, DNA methyltransferase is able to recognize exactly the same sequence recognized by restriction enzyme, but it puts methyl group as epigenetic mark on this sequence, making the DNA resistant for restriction enzyme cleavage. So therefore, the host DNA is protected from restriction enzyme cleavage by methyltransferase, while the invading foreign DNA, for example, phage DNA, it lacks these protective methyl tags and is degraded by restriction enzymes. So in this respect, the restriction modification system functions as innate immune system of bacteria that is able to differentiate between self and non-self using the simple methyl tag. So, as I said, restriction enzymes, they function as DNA scissors. They recognize short nucleotide sequences, like this one shown for ECO1 restriction enzyme, and cut it, introducing a double-strand break. And different restriction enzymes are able to recognize different target sequences. Therefore, the cleavage pattern, for example, of lambda DNA by different restriction enzymes give different bands. And in my lab, for many years, we tried to understand actually how restriction enzymes can specifically discriminate their targets. And there is a really large number of restriction enzymes. It's around now more than 4,000 of different restriction enzymes. And this is a question actually then, which restriction enzyme you take for this study. And I'm using this slide as illustration. It's kind of, it's the same problem, but anyway, so we decided to focus on a family of restriction enzymes that have a conserved CCG tetranucleotide in their target sites. And this conserved CCG tetranucleotide is flanked by different nucleotides outside of this conserved core. And we aim to understand how nature designed specificities of different restriction enzymes and how different restriction enzymes in nature are able to discriminate these external nucleotides outside of the conserved core. And to address this question, we decided to solve the crystal structures of restriction enzymes belonging to this family, and this work started when I was a visiting scientist and in Robert Huber lab, and continued for more than 20 years. At the end, actually, we were able to solve all these structures and try to correlate the structure and function of restriction enzymes, trying to understand how they recognize different target sequences. And we even were able to derive some rules. And we naively thought that if we understand actually how nature designed different specificities of restriction enzymes, so we thought maybe we can then use the same principles and engineer restriction enzymes to any target sequence, making them useful tools not only in molecular biology, but maybe also in gene editing. But the problem is that restriction enzymes recognize their targets reading the combination of donor and acceptor atoms in the major groove of the DNA. And each base pair contains a characteristic signature of donor and acceptor atoms in the major group. And restriction enzymes read these donor and acceptor atoms through the hydrogen bonds and hydrophobic or van der Waals interactions between amino acid residues and DNA, the DNA bases. And that makes reprogramming of the restriction enzymes really a difficult task, because if you would like to reprogram restriction enzyme for a different sequence, you need to actually engineer new amino acid residues that will recognize different sequence. We did these experiments, we designed some variants of restriction enzymes, but actually it was a really challenging task, because actually designed variants often showed the off-target cleavage recognizing different target, but related target sites. So at this point, I was a bit tired of our efforts in characterization of restriction enzymes. So started to look for something new, and then in 2007, a paper appeared in Science Journal that claimed that there is a new antiviral defense system called CRISPR, and this antiviral defense system provides resistance against viruses in prokaryotes. That looked for me very interesting, and because it claimed that there is antiviral defense system, but no one knew how the system works. And so when phages infect the bacteria, most of the bacteria die. However, a small number of bacteria survive. And it turned out that survivors were able to hijack small pieces of invading phage DNA and incorporate them into a so-called CRISPR array that is present in the bacterial genome. And these pieces of phage DNA are incorporated as a new species, and the CRISPR array is comprised of conserved repeat sequences interspaced by different spacer sequences. And then this CRISPR array is transcribed, generating pre-CRISPR RNA that is further processed into CRISPR RNA. And this CRISPR RNA, together with Cas proteins that are encoded by Cas genes, assemble a so-called effector complex, and this effector complex actually triggers degradation of invading phage DNA during the next round of infection. So in this respect, CRISPR-Cas systems function as an adaptive immune system of bacteria because they are able to recognize and memorize the invader in the form of spaces in the CRISPR array, and then use this memory to fight the invader. And the paper in Science that was published in 2007, it described CRISPR systems that were present in Strep thermophilus, and Strep thermophilus is an industrially important bacteria that is used for making cheese and yogurt. And in Strep thermophilus, Philippe Horvath and Rudolf Barangu identified 4 different CRISPR-Cas systems that differ by the number and arrangement of Cas genes. And of course, again, it's the same problem which one you choose for your studies. You're choosing now not from 4,000, but only from 4 different CRISPR-Cas system, and we decided to focus on the CRISPR-3 system primarily of 2 reasons, because it contained the smallest number of Cas genes, and the second thing was that one of the Cas9 gene, in fact, it contained signature residues that we seen in the active sites of some restriction enzymes. And that was a kind of indication for us that maybe this could be a functional protein. So we didn't know in the lab how to work with Strep thermophilus. We were not making yogurt or cheese in the lab. So we decided to simplify the things, and we decided to transfer the whole CRISPR-3 region into E. coli that was a workhorse in the lab. And to our surprise, actually, this transplanted CRISPR-Cas system from Thermophilus into E. coli, it worked nicely, and it provided resistance against invading phage or plasmid if spaces matching phage or plasmid DNA were present in the CRISPR array. And this was a first indication that actually CRISPR systems are transposable or mobile units that could be transferred between different organisms. And it looks like, okay, so it's not big deal transfer CRISPR system from one bacteria to another, but if you think that this is a gram-positive bacteria, this is gram-negative bacteria, and there is quite a distant evolutionary distance between them. And then when we had the CRISPR system in E. coli, so using the simple genetic tools that are available in E. coli, we were able to demonstrate that, in fact, the Cas9 gene is the only gene that is required for interference. We can kill all the genes in the CRISPR cluster except Cas9, and we'll still see the interference. So then the obvious step was just to try to biochemically characterize the Cas9 protein, and Giedrius Gasiūnas, who was at that time a PhD student in the lab, managed to isolate Cas9 protein, and it turned out that the Cas9 protein co-purified with 2 RNA molecules, and CRISPR RNA and trace RNA, and these 2 RNA molecules made a kind of partial duplex, and then we were able to show that, in fact, the CRISPR RNA molecule in the Cas9 ribonucleoprotein complex that acts as a guide that is able to recognize a complementary sequence in the DNA and bind to it, generating a structure that is called R-loop. And in this R-loop, actually, Cas9 protein is able to cleave 2 DNA strands, introducing double-strand break. So, in fact, the Cas9 protein functions as a restriction enzyme. It does the same thing. It recognizes specific sequence in the DNA and introduces a double-strand break. But the important difference is that in the case of restriction enzymes, as I described for you, the specificity is achieved through interactions between amino acid residues and donor-accepting atoms on the base edges of the recognized sequence. In the case of Cas9, actually, the specificity is achieved through the Watson-Crick base pairing between RNA molecule and the complementary DNA strand. And the simple modular organization of the Cas9 ribonucleoprotein complex, where specificity is provided by CRISPR RNA molecule and DNA cleavage is provided by Cas9 protein, actually pave the way to development of universal or versatile genome editing tools. Indeed, by simply changing the CRISPR RNA in the RNP complex, Cas9 protein could be delivered to nearly any target in the genome where it will introduce double-strand break, and this double-strand break will be repaired either through non-homology end joining or homology repair. And these 2 repair mechanisms could be used either for gene knockouts or knock-ins. And indeed, after 12, so before 2012, Cas9 was a part of a CRISPR-Cas system, and not many people were interested in these systems. But after 2012, actually, it became a genome editing tool, and all the world suddenly became very much interested in this system because actually it allowed you to generate gene knockouts and knock-ins, simply making a programmable targeting of the Cas9 RNP complex. And then Cas9 protein actually was used for gene editing in many different model systems, including humans. So this is kind of a very simplified explanation what could be done with Cas9. So you know that actually sickle cell disease is a kind of genetic disease that results due to the single mutation in the gene that encodes one of the hemoglobin subunits. And as Mendel taught us, actually, this results also to a different phenotype. So in the healthy person, the red blood cells that carry hemoglobin have the donut-like structure, while in the case if a person carries a mutation, so the red-shaped cells get a sickle-like structure, and therefore the disease is called a sickle cell disease. So in this kind of sickle blood cells clogs the capillaries, causing the manifestation of disease. So now the gene editing tools like Cas9 allow actually to reverse this mutation, and therefore, in fact, you can treat genetic disease. And I borrowed this slide from the Innovative Genomics webpage, which shows that, in fact, the Cas9 protein is now widely tested in different phases of clinical trials. And in fact, in November 2023, the first therapy in the clinics was approved, so the Cas9 was approved for the treatment of sickle cell disease, and now the same approach is also used for treatment of beta thalassemia. And you see that there are many actually clinical trials that target very, very different ranges of disease in phase 1, phase 2, and phase 3. And I guess it clearly shows you that CRISPR is now moving into the clinics. And in fact, it's really fascinating to see that actually we and others demonstrated that Cas9 protein acts as a genetic scissors that can introduce a double-strand break, and in 10 years nearly, actually we already have this system in the clinics. So it's a great system, but it still has some limitations, and there are still some challenges of the Cas9 technology. And I listed some of these challenges in this slide, and I will talk briefly in my talk about the targeting space limitation and delivery problems. And of course, if there are challenges, so then it should be some approaches that allow us to address these challenges. And in principle, there are two general approaches. One can try to engineer, for example, Cas9 protein to make to limit targeted space limitation of this protein, or, for example, make it of smaller size. And there is another approach. You just look at the natural diversity of the CRISPR-Cas systems. And in my lab, actually, we took this natural diversity approach. And the CRISPR-Cas systems are very diverse. You are probably familiar mostly with Cas9. But if you look actually at the diversity of CRISPR-Cas systems, it's really stunning. And CRISPR-Cas systems are classified in two large classes, class 1 and class 2. And the base of classification is on the structure of the effector complex that cuts DNA. In the class 1, effector complex is assembled of multiple protein. In the class 2, as I already illustrated by Cas9, the effector is a single protein that is bound to a single or two RNA molecules. And this class 2 system also exhibit high diversity, and they are further subdivided into different types. For type 2 and type 5, they act on the DNA, and type 6 act on the RNA, but all of them relies on the single protein effector. So, as I already told you, the specificity of Cas9 protein is dictated by interactions between guide RNA and target DNA sequence. And these interactions result in the formation of R-loop. However, in order to initiate RNA binding to the complementary DNA strand, Cas9 protein first has to recognize so-called PAM sequence, and PAM stands for protospace adjacent motif. So it first has to read PAM sequence, and after reading the PAM sequence, then RNA can sneak into the DNA duplex and bind to the target site. And different Cas9 proteins, they show different PAM sequence varieties. And this requirement for PAM sequence, it limits target site selection. For example, in the case of aspergillus Cas9, the requirement for GG-PAM constrains targeting to every 8 base pairs in human genome, and making nearly 75% of mutations associated with human disease non-accessible for genome editing if a precise targeting is considered. So we reason that actually maybe we can overcome this targeted space limitation if we look at the Cas9 orthologs. And Cas9 orthologs are really widely distributed, and we developed a kind of phylogeny-guided approach to identify Cas9 orthologs, and we built bioinformatics routines that allowed us to identify Cas9 sequences. But for each Cas9 sequence, the PAM requirement should be established. PAM requirement should be established experimentally. So to establish PAM requirement for Cas9 proteins, we developed also a simple pipeline that allowed us to identify experimentally the PAM requirement that the PAM that licenses cleavage of the DNA by Cas9 RNP. And then we selected nearly 100 of different Cas9 sequences present on the Cas9 phylogenetic tree, and then using this pipeline decided to characterize PAM requirements for different Cas9 orthologs. And what we found that indeed different Cas9 orthologs showed very different requirements for the PAM sequence, and we actually seen Cas9 orthologs that recognize A-rich sequences or C-rich sequences or G-rich sequences and so on, and different combinations of the nucleotides. So these Cas9 orthologs, in principle, they open the possibility to address the targeting space limitation in genome editing experiments that result due to the PAM requirements. And the next limitation that I would briefly talk about is the delivery. So the clinical trials that I described and the Cas9 clinical trials, they depend on the ex vivo editing. So it means that cells are taken out of the body, modifications are carried out in the labs, and then cells are actually put back into the body. But if we think about in vivo gene editing, so editing in human bodies, you need to think about delivery. So the question is how to deliver the gene editing tool to a specific type of cells in the body. And then, actually, one of the ways of the delivery is based on the use of adeno-associated viruses that actually show different tropism, and these different viruses could be, in principle, targeted to different organs in the body. And it's a tool that is already licensed by FDA for different applications, but it has its own limitation. The capsid for AV is quite small, and there is a packaging capacity that is limited to nearly 4.7 kilobases that you can put into AAV. And Cas9 protein is a big protein. It's comprised of nearly certain hundred of amino acid residues. And if you think about packaging Cas9 into AAV together with guide RNA, it is becoming challenging. So therefore, a smaller genome editing tools are desired. And again, to find these smaller gene editing tools, we decided to look at the natural diversity. And in this case, we focused on the type 5 CRISPR-Cas systems that were described in 2017 and 18 by Shmakov and Harrington. And these were actually CRISPR-Cas systems that had an effector protein that initially authors called Cas14a, and this Cas14a actually belonged to the phylogenetic tree of Cas12 protein, and these and now we call these Cas14 proteins Cas12f effectors according to a new nomenclature, and these Cas12 effectors, they actually belong to the 3 different branches of the phylogenetic tree. But despite of these kind of differences, phylogenetic differences, they all share 2 common features. They are pretty small proteins. The size of the Cas12f is around 400 to 700 amino acid residues. That is half size or 1/3 of the Cas9 of Cas12f protein. And they share a single concept RAFC active site that is present in Cas12a. And therefore, because they are small size, we call them miniature proteins. And then, using our PAM identification approach that I described you for Cas9 protein, we actually decided to look whether these Cas12F proteins also are able to cleave DNA, introducing a double-strand break, and whether they require PAM sequence for cutting DNA. And we selected at least 10 different Cas12F variants, and using this PAM identification assay, showed that indeed the Cas12F proteins, they are able to introduce a double-strand break in the DNA in a PAM-dependent manner. So they need, again, a PAM sequence to cut DNA. So then, during recent years, a number of papers appeared that showed that these miniature Cas12F family enzymes can efficiently edit mammalian genome, could be used in plants and other different other systems. So in this part of my talk, I just simply showed you that looking at the natural diversity of CRISPR-Cas systems, we were able to identify Cas9 orthologs that recognize different PAM sequence, and this helps us to overcome the targeted space limitation of Cas9, and also to identify Cas12F miniature nucleases that are small genome editing tools compatible with AAV delivery. So the Cas9 and Cas12, these are nice tools, but they are part of the CRISPR systems. And the next interesting scientific question was actually where Cas9 and Cas12 actually come from. And if you look at the phylogenetic tree of Cas12 protein, so you can see there are different variants or subtypes of Cas12 protein. But on the top of the phylogenetic tree, there is a protein that is called TNPB. And this TNPB protein, in fact, is a small protein. Its size is around 400 amino acid residues. And it has an active site that is similar to the Cas12F family. We call it RAFC active site. But actually, all Cas12F proteins, they actually reside close to the CRISPR array, and they function as the CRISPR system, providing interference against invading phage DNA. However, in the case of TNPB, there are no CRISPR array in the vicinity. And a few years ago, actually, we decided to look at the TNPB function and what TNPB means and what is the function that was known for TNPB. The TNPB is actually a member of IS200 transposon family, and transposons are mobile DNA that is able to jump between different locations in the DNA. And the specific transposon where we focused actually was present in Deinococcus radiodurans bacteria. And this transposon belongs to the family that I just mentioned, and it's a very simple transposon, and it's probably one of the most ancient transposons. It contains a TNPA gene and sometimes TNPB gene, and this TNPA gene or TNPA and TNP gene combination is flanked by left and right transposon elements that are actually important for transposon mobilization. And this transposon actually is excised and inserted at the specific sequence TTGAT that is just in front of the left transposon element. And it was well known for a long time that TNPA is a transposase that enables transposon mobility, allows transposon mobilization and jumping between different locations in the DNA. But sometimes TNPB is present in this transposon, And the function of TNPB was not clear. And actually how this transposon is moving between different locations in the DNA. So the transposon movement is related to DNA replication. And during DNA replication, tNPA transposase excises the transposon from one of the DNA strands during replications. And the transposon, excised transposon forms a secular single-stranded DNA intermediate. And then actually this transposon, excised transposon, is inserted into another location of the genome specified by this TTGAT sequence. So again, the role of TNPB in this mechanism was not clear. So in order to address the role of TNPB in the transposon function, we decided to isolate TNPB protein. And we expressed TNPB protein together with inactivated, with transposon with some elements inactivated to stop the mobility of the transposon. And we were able to isolate TNPB protein. And what we found that TNPB protein co-purified with RNA molecule. And then we isolated this RNA molecule from the transposon and sequenced it. And RNA-seq revealed that actually this RNA molecule originates from the gene that encodes the TNPB protein. It has part of the coding sequence of the TNPB protein, also includes right transposon elements, and then 16 nucleotides of flanking DNA. So since this RNA contained the right transposon elements, we called it RNA. So then we realized that maybe it functions similar to the RNA-guided nuclease, and then we decided to look whether TNPB RNP is able to cleave DNA, and we designed a very simple assay. We actually expressed TNPB protein together with RNA molecule in E. coli, and then actually looked at the cleavage of the DNA that occurred in the plasmid DNA library. And what we found, indeed, that this TNPB protein was, RNP complex, was able to cleave DNA, double-stranded DNA. But actually for the cleavage, it required a specific sequence, TTGAT, to be present in front of the target sequence. And if we look at the transposon, so it's exactly the same sequence that is required for transposon mobility. And we have shown that TNPB cut double-stranded DNA, introducing double-strand break, and it had several cleavage sites on top strand and a couple of cleavage sites on the bottom strand. So, in fact, these experiments showed that TNPB, in fact, is RNA-guided endonuclease, very similar to CRISPR nucleases that I described before. It contains a RNA that acts as a guide RNA, and then it contains a TNPB protein that does the cutting. So, but the cleavage site for TNPB is composite. It's comprised of TAM sequence, or the transposon-associated motif sequence, that is located in the actually left side of the transposon. And then the other part of target site is actually recognized by RNA that is complementary to the other side of the transposon. So when transposon is present in the DNA, so the target site for the TNPB nuclease is interspaced. But when actually transposon is excised from one of the DNA strands, so then actually the TAM and sequence recognized by the RNA, they come together, making a cleavage site for TNPB. And then cleavage, and then TNPB can cleave this DNA. So the question is how now this TNPB endonuclease fits into the transposon mobility mechanism. As I said, during the transposition, the transposon is excised from one of the DNA strands. So, in fact, it's excised. It's excised from the lagging DNA strand. So, in the next DNA strand, the transposon is still present. So, when the transposon is excised from one of the DNA strands, so then the target site for TNPB is reconstituted, and TNPB is able to cleave the target site in one of the DNA copies, and then this double-strand break triggers homology recombination that results in the what's going on there? Results in the reinstation of the or reconstitution of the transposon copy in the another DNA copy. So in fact, the TNPB plays a kind of safeguarding role in the transposon mobility. In the transposon mobility. So when the transposon is excised from one of the DNA strands, it could be lost, because if the insertion event was not successful, so then the transposon will be lost. It will be not inserted anywhere. And then, actually, after replication, you will have two different DNA copies. One of the DNA copy will contain a transposon. Another copy will lack the transposon. And after cell division, so then it will be a cell population which actually lacks the transposon. So the TNPB, in fact, safeguards, provides a safeguarding mechanism that allows actually to reinstate the copy of transposon in the case if it is lost. So then, since we demonstrated that TNPB acts as a functional programmable RNA endonuclease, an obvious next step was just to look whether it can cleave DNA in eukaryotic cells. And what we found that indeed, TNPB was able to introduce indels at different target sites with different efficiency, in this case in HEC cells. But anyway, without any modifications, indel efficiency reached up to 20%. So it shows that TNPB is emerging now as a novel genome editing tool. So, of course, the next interesting question was, and how the TNPB complex looks like. And to address this question, we in fact solved the structure of the TNPB complex, the binary complex, TNPB bound to RNA, and also a ternary complex, TNPB bound to the DNA. And what we found that indeed, TNPB complex revealed that TNPB protein is comprised of two different lobes, rec lobe and NUC lobe. The active site resides in the NUC lobe. Rec lobe is actually required for binding and recognition. And actually, it also has a long RNA molecule, RNA molecule that is bound to the protein. But anyway, this TNPB variant is really a small one. Its size is only 400 amino acid residues. So there are many structures of different Cas12 variants solved, and in this structural tree of Cas12 variants, TNPB is located here on the left. And actually, if you do structural comparison, so the TNPB protein is closely related to Cas12F family miniature nucleases of the CRISPR-Cas systems. And actually, TNPB, but in fact, the original Cas12 protein actually is very different from TNPB. It has really many protein insertions in the kind of concept core TNPB. But it's interesting that in the case of TNPB, we have a small protein and large RNA molecule. In the case of Cas12a, we have a large protein and a small RNA molecule. So it looks that during evolution, protein was able to overtake some roles that are taken by RNA in a small TNPB variant. But anyway, in any case, this structural comparison revealed that TNPB is in fact a minimal structural and functional core that give rise to the Cas12 protein family. So then it was somehow, and Eugene Koenig explains that somehow TNPB occurred close to the repeated DNA sequences similar to CRISPR, and then this is how CRISPR systems developed. So another thing what we did, we looked at the sequences of TNPB, and actually the TNPB protein is one of the most abundant proteins in bacteria. There are millions of different TNPB sequences, and what we found that actually all other Cas12 family nucleases, they are distantly related to TNPB family that makes a core of the sequence comparison. And this also showed that TNPB is evolutionary predecessor of the Cas12 family. So far, I was talking about antiviral defense systems that function through the cleavage of invading phage DNA. I talked about restriction modification systems and CRISPR-Cas systems, but are there any other strategies for antiviral defense that are not related to the DNA cleavage? In the couple of next slides, I will show you another antiviral defense system that we described recently. So Rotem Sorek actually recently described a new Torres antiviral defense system that was identified in Bacillus cereus. And this is a simple antiviral defense system. It's comprised of two genes, THS-A and THS-B. And then THS-A is comprised of two different domains, SIR2 domain and slug domain. And THS-B just contains a single toll interleukin receptor domain. And during his biochemical studies, Rotem was able to show that, in fact, in response to fate infection, that THS-B is able to recognize invading phage DNA by the mechanisms that still has to be determined. And in response to that, it synthesizes a new signaling molecule that is from using NAD as a precursor. And this cyclic molecule is a cyclic derivative of ADP ribose. And this signaling molecule, in fact, is recognized by THS-A protein that acts as a factor. It binds the signaling molecules and then actually triggers NAD degradation, resulting in a growth arrest, because NAD is a key metabolite in cell, and if you deplete the cell of the key metabolites, so the cell will go into growth arrest. So, but how the THAS is activated by this signaling molecule was not clear, and we decided to address this question and by trying to solve the structure of THS protein in the presence of THS-B and NAD. And what we found, and we used a cryo-electron microscopy for that, and what we found that actually long helical filaments of THS. And it turned out that these long helical filaments are formed by THS tetramers that are stacked onto each other. And this helical core is made of the SIR2 domains in the middle, and slug domains are exposed outside of the helical core. And these slug domains are, in fact, the sites where these signaling molecule is bound. And when signaling molecule is bound to this lock domain, there is a conformational rearrangement of CIR2 protein that in fact is a NAD degradation enzyme that is able to degrade NAD. And when these filaments are formed, so then actually NAD's activity is significantly increased. It looked like an interesting model based on structural studies, but what you can do to actually confirm this model, so we identified the contact residues that are important for filament formation. And we mutated these contact residues, introducing alanine replacements. And what we found that using a DLS technique, what we found that actually introduction of interface mutations important for helical formation actually disrupted helical formation. And exactly these mutations also disrupted NADase activity of the THase. So taken together, this experiment allowed us actually to propose the following mechanism of THS activation. So when phage infects the cell, so THSB actually senses the invading phage, and then actually it synthesizes this signaling molecule, and this signaling molecule actually binds to the THase effector that oligomerizes, and then actually this oligomerization triggers the NAD degradation activity that results in the growth arrest. So this example shows you that antiviral defense systems can use very different strategies to cope with phage infections. And why these antiviral defense systems could be important? So we already talked about restriction modification systems, and during 1960s and 70s, restriction modification systems actually allowed to develop recombinant DNA technology that finally culminated also in a Nobel Prize for discovery of these restriction modification systems. So in 2010 and 2020, CRISPR-Cas system, actually, that is also antiviral defense system, resulted in development of genome editing technology that, again, culminated in a Nobel Prize. And so now, actually, hundreds of new antiviral defense systems are identified. So the question is, what awaits us there? So I guess that we only looked at the tip of the iceberg. So many interesting things are still below the water. And I think that this kind of studies of these antiviral defense systems could reveal new chemistries, new biochemical mechanisms, and also new applications. And finally, I would like to acknowledge people from my lab who contributed to different parts of this work, our collaboration, Guillermo Montoya and Arturo Carabias, with whom we solved the structure of TNPB. And also, I would like to acknowledge our industry collaboration, the Kazyme company that we established in Vilnius, and Gedros Gasiūnas, who was a former PhD student of mine, is now COO of Kazyme. We have a long-standing collaboration with Corteva company, developing together genome editing tools for plant applications, and we have also collaboration with New England Biolabs. And at this point, I would like to stop and thank you for your attention.
Yeah, thank you. Thank you for this exciting talk and for giving us this interesting overview of all the mechanisms that bacteria have to fight against viruses. So the talk is open for questions now. Yes.
Thank you for the talk. I would like to ask, what is actually the biological role of the PEM motif? Why is it needed to have it there for the bacteria?
Yeah, it's a good question, and it's an important question. The PEM sequence, in fact, is the element that allows bacteria to discriminate between self and non-self DNA. In the invading virus, there is a DNA sequence that is complementary to CRISPR RNA, and then there is a PAM sequence in the vicinity of this sequence, of this complementary sequence. And this enables DNA cleavage. In the CRISPR array, there is a DNA sequence that encodes only the guide RNA that is complementary to DNA, but in the CRISPR array, there is no PAM sequence. So it means that bacteria cannot cleave its own DNA. And this protects bacteria in a bit different mechanisms that I described for methyl transferases in restriction modification system, but it actually acts as a system that allows bacteria to avoid autoimmunity, not to cut its own DNA.
Thank you for a great talk. I was just wondering, I should know the answer probably, but are there any phages that contain RNA, and then is there a defense mechanism in bacteria against incoming RNA, like in mammalian cells?
Yes, there are phages. There are some phages in bacteria that are RNA phages, and then there are CRISPR-Cas systems that are targeting specifically RNA phages. So they recognize, instead of recognizing DNA, they recognize RNA and then actually have another effector protein that is able to destroy invading RNA. So that's kind of bacteria arsenal for antiviral defenses is very well developed, I would say. Yeah.
Also very interesting talk. My question would be in respect of the phage defense system, or in a way, can it hijack the low efficiency of the DNA repair, or actually improve the repair of the cleaved phage DNA or RNA to get the resistancy?
Yeah, so I don't have an exact answer, but the double-strand break repair by non-homologous end joining is kind of not very popular in bacteria. So there are some bacteria that have it, but most of them don't have it. So in the case of bacteria, so if there is a double-strand break and if there is no homology copy, so then actually it will be bad. But I think that in eukaryotes, what we see is that sometimes the efficiency of Cas9 on different sites differ, but it could be also that actually we have many productive cutting events, but also we have many repair events. So, and only some slips results in the indels, and most of these double-strand breaks are repaired. So if you can play with this kind of, maybe you can affect the outcome of editing, yeah.
So you described actually also these other systems beside CRISPR, like transposons and Toris system. So, simple question, do you expect that these tools will also trigger such a boom like CRISPR-Cas9 system, that they will be now re-engineered into like genome editing tools worldwide?
You never know what will happen when you are doing your research. So when we were studying Cas9 as a part of antiviral defense system, it was an interesting protein, but actually we didn't expect that, we studied the system because it will have some interesting applications. So I think that in research, things are going in certain order. You are doing basic research, trying to answer very basic biological questions. And when you understand biological mechanisms, so then you can start to think about how to apply these things. Because actually, if someone would ask me to design a genome editing tools from scratch, that would be a challenge. But when I know how Cas9 works, so from my biochemical studies, so then I can think about, okay, what could be a promising application for this tool? So, and the same is probably true for these new antiviral defense systems. We still need to realize what kind of chemistries are going on, because I just described the antiviral defense systems that depletes NAD. But there are antiviral defense systems that depletes ATP. There are antiviral defense systems that make holes in a cell wall. And these holes actually results in the killing of the DNA. So kind of, it could be very different applications, but initially we have to understand how these systems work.
Yeah, absolutely, I agree.
Hi, do we know if the PAM sequence is recognized in a similar fashion as the restriction enzyme recognizes the major groups of the DNA, like receptor, donor, and if it's sensitive to DNA methylation?
Yeah, so the mechanism of the PAM sequence recognition is very similar to the mechanism that I described for restriction enzymes. There are amino acid residues that recognize donor acceptor atoms on the base edges, and then actually this recognition often results in the DNA deformation, for example, bending DNA. And that actually bending could serve as initial step for RNA binding, because then DNA is pre-opened and then RNA could sneak in and try to extend the hybridization further. So, and therefore, since the rules are the same, I don't know how many of you worked with restriction enzymes, but you know, if you're doing DNA cleavage and you add too much of restriction enzymes, you see cleavage at sites that you didn't want it to cut. And this is because the concentration of restriction enzyme was increased, and then actually you exceeded the KD that is required for binding for these non-specific sequences, and then cutting results. The same is actually also with PAM recognition. Sometimes actually increased expression of Cas9 protein can force the Cas9 protein bind to undesired PAM sequences and trigger DNA hybridization, RNA hybridization, yeah.
Are there any further questions? If not, then I think I can close the session. Thank you again for the great talk. Thank you all for attending.
Thank you. Thank you. Oh, thank you.